I want to compliment Murat for his leadership in organizing this conference when he invited me to speak at the first of these 4 years ago. And in each conference since, the rationale has been how does computing learn from brain circuitry to go beyond the von Neumann paradigm, to go beyond the capabilities of computing that we've seen become limited by physical limits, and draw from brain circuitry to go far beyond that, both in power consumption capabilities as well as in computational capabilities. You know, look at a hummingbird function. What's the size of that circuitry and the power, and look at what a hummingbird does. I don't think a convolutional net, even though Amir's spectacular talk and what his company is doing is just going to be breaking the back of so many commercial problems, and there's many, many years of runway that they and their competitors have. I don't think that they're on the path of making a sensory motor system like any of those that are in nature. So the charge of this meeting and its goal is to draw from neural biological systems to extract, as the meeting's title refers to, neurally inspired computing elements. And so for that, what I wanted to use this talk for, because it's fairly short and because I think it gets lost in the success of deep convolutional nets, I wanted to put into context the history and the way forward and the point that we are in that history now. From the 40s with McCulloch and Pitts and classical AI, to the more neural systems that Rosenblatt and actually Turing introduced, believe it or not. Alan Turing, 1948, this is an not widely published manuscript that he submitted to the British, some unit of the British Army, where he first conceived of what is on the left, neural nets. Rosenblatt's perceptron is much more familiar in the late 50s. Minsky and Papert killed that, but it got resurrected by Hopfield, as we all know, in the 80s. And in the 80s, in fact, in 1980, a fellow named Fukushima, how many people have heard of the neocognitron? Pretty good chunk, maybe 25, 30% of the audience. So in 1980, Fukushima published one of several lucid papers developing a system he called the neocognitron, which has been made famous and extended greatly by Jeff Hinton and other colleagues of his 30 years later, and that's the convolutional net, which there's the Biological Cybernetics 1980 landmark paper, and here's the system, and if you look at it, you'll recognize a convolutional net. It interchanges sensory feature layers with pooling layers, like simple cells and complex cells, and it iterates that in a deeper and deeper hierarchy. You can see it's convolutional on the top panel, in the sense that the word convolutional implies. And that's now been extended by our friends at Google and other places. You know, if any of you have visited Google, there's just such a fabulous, let's go do it spirit about that place. And here they've just gone and done it with, you know, now 25, 30 layer convolutional network. The convolutional feed-forward layers are in blue. The pooling layers are in green. Softmax is a form of soft winner-take-all, of course. And they've just, as Tom Dean of Google says, this is just the neocognitron and the 1980s distributed processing, but it's just done right, done with a vengeance, scaled up. And here Amir, where's Amir? There's Amir back there. Amir has just brought us the current instantiation of this being applied to 25 different technical problems. Chris Bhaskar will tell us about the 26th technical problem that it can be applied to this afternoon. One thing that this audience should be aware of, even though it's good and bad, is that the power of that simple paradigm of deep convolutional nets wrenched into extraordinarily high-performance applications by Google, by Nirvana, by others, that's going to bring value to solving a lot of real-world problems, and we've got to be careful that it doesn't suck all the oxygen out of the underlying, because it is going to be working so successfully, out of the underlying intellectual opportunity, which is to draw from brain circuitry. Christoph showed us all the extraordinary depth and complexity of real neural circuitry. He's got 310 people hard at work every day unraveling it. So how do we bridge that gap? How do we draw from neural circuitry without getting lost in 10 miles of detail and yet not satisfy ourselves with the real simplicity of convolutional nets? And again, they can do great things, not only identify separately cats and dogs, but even recently, profound capabilities that you might consider action selection in a complex environment, which is playing Go. I mean, I think this is really something to be reckoned with. This paper that came out about 5 weeks ago that I'm sure most of us have heard about or read, which takes convolutional nets and beautifully extends them. I mean, even a simple paradigm can be extended not only to 30 layers, but can be extended in concept. So here, I won't go through that paper. Many of you have read it. But they take 13-layer nets, one for action selection, first trained with supervised learning and then retrained with reinforcement learning. They have a separate net called a value network that learns the likelihood of a win from any given position. And then they just drive this with training and extension. And 6 weeks ago, they beat, in the game of Go, they beat the European champion. So that's going to take neural computation and define it for the next 10 years, and a fair part of our careers, if some of us don't try to point to, define, and prove the value, the capability of pursuing real neural computation, which in many ways is different. So let me try to go back to the title of this conference, Neurally Inspired Computing Elements. And what I really wanted to do with the rest of my talk is to define what neurally inspired computing elements are. Here is the computing element of the Google ImageNet, or the DeepMind nets. This is a thresholded or sigmoid output conversion of weighted inputs to a nonlinear output function. It's essentially a single node. There are other operations, including the softmax and perhaps other normalizations that are available. But on the right, if that's the unit on the left in a cartoon schematic, on the right, as Christoph has teams studying night and day, on the right is a model of that thick trunk layer 5B neuron that Christoph mentioned. So what does it look like? If we're doing the reverse engineering of a Pentium, from another planet, we get a sample of a Pentium, what are we going to do? We're going to understand transistors and how they work, and then how they can be combined. So what I'm going to point out is that these 5 characteristics of neural circuits and their components, those are universal. They're not just found in some particular application, in some particular species for some particular sensory motor purpose. Every one of these 5 has evolved and is present in every neuron in the cortex of the brain of not only all of us, but of every creature, and is used for every function. So these are universal computing mechanisms that are ubiquitous, and we should at least be thoroughly familiar with them as we think about neurally inspired computing and its development. So neurons are branch structures. They're not properly thought of as single points. Those branches are nonlinear. They integrate inputs in ways that are not simply summing and firing, but much, much, much richer than that. in ways that we could spend hours talking about. Those local events not only amplify or suppress combinations of temporally coordinated input, but they also gate wiring. But the inputs that we're talking about, those inputs, the inputs in Google ImageNet are multiplicative weights, and that's why Nirvana needs to have matrix multiplication accelerated so importantly. Well, real synapses are very different. Real synapses are probabilistic in their transmission, and that probability dynamically changes on the 10 millisecond, 50 millisecond, 100 millisecond level as a function of patterns of input. So they're temporal pattern filters. And that function, as I'll show at the end of the talk, that function, that capability, that the dynamic changing of probability of transmission is central for their function. And it's different in every connection type, and it's present in every single synapse in your brain. That nature of that transmission, that it's probabilistic and that the probability itself changes. So how can we possibly conceive of neurally inspired computing that doesn't incorporate it systematically? All of these elements, the dendritic excitability that's nonlinear, the degree of excitability, synaptic transmission probability, all of them are locally homeostatically modulated. This is a universal, ubiquitous characteristic. It's necessary for the function. It wouldn't be universal if it weren't necessary for the function of biologically evolved neural computation. And lastly, as I was bringing up with Christoph towards the end of during the questions, these circuits, it's self-evident, but it's important to say these circuits wire themselves. They wire themselves during development prenatally. They then wire themselves further during sensory motor experience. That self-organization of wiring is pivotal to the function of neural circuitry. It's not just weight modifications. It's wiring changes. It's not just supervised wiring changes that are imposed, as in the GoNet, by stochastic gradient descent, sort of a global function. That's not biological, physical. That's not local. But the wiring of the circuits themselves self-organize during sensory experience. So how does that happen? We need to know that in order to build and synthesize neural circuits that would go into the future generation of machines. And since I think GoogleNet and convolutional nets are going to occupy everybody for the next 10 years outside this room, we're going to have to wait 20 years for those truly neural machines. But let's at least know what real neural elements look like. So this is that layer 5 neuron. Here I'm just injecting current into the cell body. You can see that it's a complex system that propagates information. The voltage is coded in color, of course, and that propagation of information allows the fact that the cell fired to be communicated to the distant reaches of the cell, for example, to gate what's called, everyone knows what Hebbian plasticity is, the conjunction of input and postsynaptic spiking. Well, you have to convey the information that the postsynaptic cell spiked. And so that back propagation is found in every neuron in the cortex. All those 42 different types that Christoph has. Have you found any where there's no back propagation? Probably. Yeah. So we found them in reptile cortex. It's not just humans. It's not just mammalian. Okay, so that's not how input comes in. That's how input comes in some physiology experiments, but really input comes in via synapses. So here you've got input coming into the branches. It doesn't come into the cell body. Input comes into the branches, excitatory input. But here what you see is the complexity of electrical events in the branches. This is what happens in the neurons in biological circuits. Synaptic input comes into the branches. Those branches nonlinearly combine the inputs, and they produce events sometimes localized to the branches that contribute to firing the cell, but also gate changes in the branches, gate wiring changes, gate synaptic weight changes. So right now, right now we're looking at a computational unit. That's the transistor, that's the sort of structure called the neuron. You know, it takes 3,000 differential equations to simulate such a neuron at a intermediate level of accuracy. you can just imagine the functional space, the function of functions of the way that such a neuron integrates spatiotemporal patterns of input. But those are the computational units in biological circuits. Now, as Christoph pointed out, I think I saw this image in one of his slides. This is from our mutual friend Alex Thompson. This is just the excitatory cells. What I'll just say to echo what Christoph said is that the integrative characteristics of each of these different cell types, the 42 that he's identified, they're all different. They have different flavors, they produce different spike output patterns in response to different patterns of spatiotemporal input. But here's one thing I really want to get across, because it's surely not integrated into any aspect of convolutional nets. Perhaps one day it will be. And that is the nature of the synapses, the nature of the connection. So this is as everyone probably knows, most of us, this is a slice of neural tissue at the electron microscopic level. That thing, if you just toggle that's a two-dimensional slice of a three-dimensional object, a synapse. On the top, you see those round things, those are called vesicles. On the bottom, that's the presynaptic, that's the axon. On the bottom is the dendritic spine that's receiving that input. So that's a cross-section of a synapse. Now, this is a cartoon of such a cross-section. These cartoons are never terribly accurate, but the point here is all of those white spheres, those are vesicles, and those that are adjacent to the membrane at the bottom there and fusing, those are the ones that when they fuse, relay transmission. So the long known, 50 years known, but really only in the early 90s, even in neuroscience, was really talked about again in neural computation, is that transmission is when the axon fires, it doesn't mean that the synapse necessarily relays input. The transmission is purely probabilistic, it's stochastic. So here you have these two vertical lines are the presynaptic axon firing, and the postsynaptic response in a voltage change, or actually a current change, is what you see here. And you see sometimes the axon fires, but there is no throughput, and then the next time there is. And this is a stochastic process. So there's a probability of transmission upon invasion of the axon. And what's really remarkable is that probability changes with use. It changes on a very rapid time scale. This is a 40 hertz signal. These are the averaged postsynaptic effects of the thalamic cell here in that black dot going to this stellate cell in layer 4. It's an excitatory cell that receives the initial thalamic input. And if you this is the average of 250 trials. The individual trials are stochastic. So this looks smooth and regular, but it's not. So this is the probability of transmission, called P in the literature, of the input from the thalamus to that regular spiking cell when the input frequency is a regular 40 hertz input frequency. If the input frequency was 80 hertz or 20 hertz, this fall off would be different, because that fall off is a dynamic thing that's a function of input. And it can be well modeled. Well, the richness of the circuit becomes apparent when you look at the input of the same thalamic cell to the interneuron counterpart in layer 4, the fast spiking interneuron. Here is the average. You see how those postsynaptic voltage changes are narrower. That's the nature of interneurons, is that they have rapid time constants. But you see also that the average EPSP, the average response, is really going down enormously. This is because the depression in not the weight, but the depression in the probability of transmission is more profound with the input to the interneuron than the input to the excitatory neuron. Okay, well, so what is the implication for the circuit? Well, this is the world's simplest circuit. It's just the thalamic relay cell plus 2 cells in layer 4. This is 2 out of 42 in the cortex. But what's going to happen here? Well, at 40 hertz, the initial input going into the stellate cell and the interneuron may be comparable in weight, and they'll both be activated. Of course, it gets more complicated because they interact with each other, and those synapses are different. But just to start with these first two, But after 3 or 4 spikes of continuing input, the input to the interneuron dials down. The input to the excitatory cell's dialing down, but not as fast. So as a result, the balance, the difference between the activation of the excitatory cell and the inhibitory cell shifts significantly during 3 or 4 inputs over, what is that now, a 75 millisecond period. And so then you get a shift in the throughput of the circuit because the interneurons are no longer gating shut throughput through layer 4 because the input to them has dialed down more rapidly than input to the excitatory cells. Well, I don't have to prove to you that if you then iterate this conversation through all the different circuit elements of layer 4 and of the neocortical column, you've got a wickedly complex, beautifully complex, because all those synapses are stochastic and all of them have dynamic changes in probability that are dependent on the type, the identity of the target. Well, that's, okay, too complicated that no one understands how that system works. No one understands how that circuit works. I did my PhD thesis on exactly such a circuit. So whatever I learned is in there, but it's only 1/1,000,000th of the whole story. But that is exactly how biological circuitry is built. It evolved that way. It didn't have to evolve that way. It didn't have to be probabilistic transmission. Could have been 100% reliable. I'll tell you at the end of the talk one example for the purpose of this depression. How much more time have I got? 5 minutes. Well, then I'm not going to tell you about homeostatic regulation, except just look up the papers of Graham Davis at UCSF. He's got 40 of the most beautiful papers on 25 different mechanisms of homeostatic regulation and their biophysical basis. And so there's homeostasis of synaptic release, homeostasis of synaptic weight, meaning what's homeostasis mean? It means that all these components adjust themselves so that the average level of activity is fixed. The average level of cell firing is fixed. And why? Because sparse distributed computation is a more effective mode than either overactive or highly silent neural circuitry. So throughout the system is woven necessarily, obligatorily, is ubiquitous homeostatic mechanisms. The last thing I'll talk about is what I was alluding to with Christoph, which is how do you wire such systems? Neural circuits wire themselves. So this is, I'll just show a little bit of this movie since time is running out. This is a system with the bottom is a 4-layer retina, 4 different retinal ganglion cell types. Then layer 4 and layer 2, 3 of the cortex. Each of these simulated cells has, of course, dendritic trees that integrate input in their dendrites in a nonlinear way. Each of these cells is connected with synapses that have different dynamic change in probability of transmission. And what you do is you play this movie for 1.5 million time steps. You could do it with simple geometric images here. It's easier to understand what the retina is doing with that. But we also used movies of objects floating around on background. And then if you integrate a set of rules for local modification of not just synaptic weight, in fact, turn off modification of synaptic weight and just have modification of synaptic stability, gated by branch spiking, neuron spiking, axon input, increase and decrease of synaptic stability leading to synapses breaking, what do you get? What's the result? So this is the result of that simulation when you use natural movies. What I have here is the set of neurons in the retina connected to layer 4 cells after, I don't know, I think this was 500,000 time steps of playing a movie to it. And so what you see here is not the receptive field. This is not the receptive field. This is the connected field. These are the cells that are connected. Of course, they lead to receptive fields that look perhaps something like V1 receptive fields. The different colors are the different branches of the stellate cells. The point is that real neural circuitry wires itself, and that's just as ubiquitous and just as central in neural computation as everything else that I mentioned. Okay, so let me quickly just close by showing you a sensory environment. I showed this. This is from actually my talk in last year's session. That's a convection field. This is an olfactory environment with 4 different olfactory sources. This is on the right here. If my mouse is working, you can see the sensory activity. It's quite complex because we've got 4 different odorant sources, and the job of the animal is to determine which odorant is it close to, where's the banana, where's the poison. So the neural problem is to take this sensory information and in the presence of an unknown background and determine what object it's near, and does it want to eat it or does it want to move away from it. Canonical, simpler, not 42 cells, but 7 different cell types in the piriform cortex. That's why I always argue that that should be added to the work program. If we then synthesize a piriform cortex circuit with these 7 different cell types, we want to predict this. This is the actual odorant concentration of the 4 different odorants. On top is the prediction. You see that they match pretty well. This is the cross correlation. So that's the fitness metric. Okay, let me now, and I'll close by saying this. What is the function of that synaptic depression? We talked about how all these different synapses are not deterministic, but they're stochastic. Why are they stochastic? That short-term depression, that means the probability goes down briefly, then comes right back up. That can only be implemented biophysically if a synapse is stochastic, or perhaps best implemented that way. We could debate that later. Well, this is comparing performance to a 4-layer convolutional net. The neural system performs better. In red is the performance of the neural system. In blue, in this dashed line is the performance of the convolutional net. Now, if Google trained it, maybe it'd be a little better. But what happened here? 2 things I just want to show you. The homeostatic set point, how active do cells modify their excitability to get to, is a parameter in the system, and it evolved to 9 or 10% in these simulations to optimize performance. That's how active cells actually are in the piriform cortex when odorants are presented. But this last thing regards to synaptic depression. This black curve traces, we started out the simulation, and we evolved these parameters in the sense of artificial evolution with no synaptic depression in the feed-forward synapses and one of those elements in the circuit. No synaptic depression. But every single time, synaptic depression evolves. In other words, the parameters that control the synaptic depression turn on, and it gets to a high level of synaptic depression always in order to optimize the functioning of that circuitry. Why? Well, heuristically, it's because it's helpful to null background in order to recognize something. So the point is, just to close, the point is that these are the elements of neural circuitry, those 5 that I enumerated. Branch neurons with nonlinear integration, with dynamic synapses, homeostatic mechanisms throughout that wire themselves. All of that, none of that is really incorporated or studied very deeply with convolutional nets, even though those are extraordinarily powerful and will be very valuable. And if we really want to pursue neurally inspired computing, I just want to commend some of us to in a systematic, quantifiable way, to integrate and harness those sorts of neurally inspired elements that are in fact found in biological circuits, to build things that work, to build machines that perform in sensory motor environments. I hope I think that's one of the rationales for this conference, and I hope that we get somewhere with it. Thanks very much.
How the homeostatic target levels are set in the model that you have shown?
Would you say that again?
How the homeostatic target levels are set in the simulation that you have shown? Are they preset as parameters, or are they computed automatically?
The homeostatic parameters, meaning the level of activity that the neuron adjusts itself to achieve, those are themselves metaparameters that are evolved in the sense of artificial evolution. Those parameters were evolved in that example.
So there is an adaptive rule that basically update this target level?
Not an adaptive rule, but rather, I could explain later what artificial evolution refers to, but it's, we evolved that metaparameter, how much activity is desired. We evolved that. Let me just get one from Thomas. I'll explain better.
So maybe very quick.
Yeah.
Yeah, very, very quick one. Thank you for pointing out that brains are not convolutional nets, but brains have evolved the way they are for different, lots of different reasons. One of them is that they perform very well, and one of them is that they had certain hardware available that they can use. How do we decide which one's which?
Yeah. So I would say that the fact that synapses could be deterministic, but they're highly probabilistic, indicates that the probabilistic transmission is there in order to enable some computational function that is necessary. But that always has to be asked, which is a physiological detail and which is a computational necessity?